I am René Bove, and it is my real pleasure to introduce today Professor Tom Higham and from the School of Archaeology and Dr. Susana Carvalho from the School of Anthropology, both at the University of Oxford, for today's School of Archaeology public lecture to discuss the fascinating topic of how science is revealing a new story of our origins. Dr. Susana Carvalho is an associate professor of paleoanthropology. She's a primatologist and paleoanthropologist, which is a rare combination in our field. And she has been one of the key founders of the field of primate archaeology. Susana has done extensive research on the origins of human technology, focusing on the behavior of chimpanzees in West Africa, but also excavating some of the oldest stone tool sites in the world in the Lake Turkana Basin in Kenya, and more recently leading an interdisciplinary research project in Gorongosa National Park in Mozambique. Professor Tom Higham, Professor of Archaeological Science at Oxford, is also the director of the Oxford Radiocarbon Accelerator Unit, and his focus has been on radiocarbon dating, the chronology of key archaeological sites, but perhaps most importantly is on what this chronology of sites intertwined with molecular studies can tell us about our evolution, especially about the later stages of human evolution. Tom has worked extensively, if you look at his published record, pretty much across the entire planet. I'm not sure I've seen any publications from Antarctica, but you can correct me if I'm wrong. But extensively in Eurasia, as well as in Africa and the New World, largely focused on radiocarbon dating. But some of the key topics that he has been addressing most recently deal with the disappearance of the Neanderthals in Western Europe, a very fascinating topic. Also deciphering the enigma of the very mysterious Denisovans that we know mostly from sites in Asia, as well as the human colonization of the Americas. But most importantly, I think, is how chronology intertwined with genomics can help us understand the later stages of our evolution and how we have become the species that we are today. So I'm very much looking forward to this discussion. Just a couple of housekeeping points. One is that this session is being recorded. And second, that if people in the public, if you have any questions that you would like to ask, please post these on the chat section and we will try to get to them at the end of the session. So I leave you with Tom and Susana.
Thank you so much, René. Thank you, René. That was great, very kind of you. And so I suppose I'm going to start, Tom.
Yep.
And let me start by saying thank you very much. It's a great pleasure, and this is for Tom and everyone. So hi, everyone, all our audience at the moment that has logged into Zoom. What a great pleasure to be here today. I'm really excited also about this session, and like everyone else in the audience, I'm also curious to know more about Tom's new book. Let me start by saying what a milestone book, Tom. I think, thank you so much for creating something that is, exciting, accurate, accessible, and entertaining or funny even. So I think sometimes we underestimate how difficult it can be to write a book that people actually read and find difficult to stop reading while they are reading. I think especially if you are a scientist that wants to talk about complex techniques and methods, like as it is your case, and explain jargon, which, I think it's really, any jargon that is not dinosaur related, I think it's not very welcome, generally speaking. I think that's where most people draw the line. Yeah. And so you focus on this time frame, on this period, where we think modern humans are around, say, 300,000 years old to the present. And this period has been completely, burgeoning with discoveries over the past 20, 30 years. I mean, it's literally the case, and you say this, that when you went a lecture on, say you're teaching about Neanderthals, you get out of the lecture, you open your laptop and discover that your lecture is outdated since the day before. And today, looking at, at the chronology of discoveries of species, this for the whole hominin lineage or clade, it was very interesting to see that we have, what was my calculation? So we have now 24 species of hominins, give or take. 9 were found in the first 220 years, and 11 were found in the last 26 years. I mean, when I say found, I say, named taxonomically, found and described. So, I mean, this says a lot about the pace of discovery, right, recently. So I think really your book reflects beautifully your different strengths and talents. If I may say, I think you clearly show, deep knowledge as a scientist and the director of the lab you have been running for such a long time, where a lot of the key work to date in some of the most important discoveries of the past 50,000 years were made and are linked to your lab, but also as a field worker, which is something I know less about in terms of your work, and also as a player in these large teams that need to work together to make these discoveries and have to turn them into this cutting edge scientific knowledge. So this is all reflected extremely nicely in the book. And it's, some of the discoveries and moments are revealed also with great sense of humor, I thought, and passing to the reader the real emotions and excitement of the discoveries that, people tend to forget. We don't happen to experience those every day. And when we do, they are sort of impossible to forget. And all of that is, it's remarkable in the book. So I absolutely recommend the book, which, I know this is not supposed to be a moment to recommend, but I do recommend. I think it's obvious it's a major success around when you see and read the reviews are raving about it. And so it's wonderful. So I thought we could start, if that's okay, by asking you precisely to tell us more about what your book, The World Before Us, is about, and why were you motivated to write it?
Well, thank you very much, Renee and Susana, for such a great introduction. It's really wonderful to be here. I've been looking forward to this for ages. And I'm really glad that we scheduled it for now, because of course things are starting, at least in the UK, to open up a little bit, and students are coming back, and it seems as though we're almost, in this part of the world at least, on the road back to recovery. But anyway, it's a real pleasure to be here and to talk to you, and I'm super excited about it, as you can probably tell. So I initially started writing this book. It's weird because, I'd never written a book before. This is the first time I've ever done it. Most of my work is in the form of short papers and so on, and getting out new results through publication and journals. So this was the first time I've ever done this. But I was actually on a lecture tour in my native New Zealand, and I was doing about 10 lectures in about 10 days. So I was traveling around a lot and giving the same lecture every time. It just about sent me mad. Actually, it sent my handlers and helpers a bit mad too in the end, I think. Anyway, what happened was that a lot of people came up to me afterwards and asked me questions, and they would say, is there a book I can recommend on Denisovans? Because I was talking about Denisovans a lot. And of course, we only discovered Denisovans in 2010. And so not surprisingly, there isn't a great deal of literature about them. In addition, we only have 4 or 5 fragmentary human remains of Denisovans. So they're not, it's not easily able to write a lot about Denisovans. So I get naively thought, I should try and write a book about Denisovans. So I launched into it, and I got about sort of a third of the way through what would be a book when I realized that it was going to be quite a short piece of work. And my editors and my colleagues suggested that perhaps what I could do was widen it a bit, because of course Denisovans are one strand of a very increasingly complicated picture and a complex picture of this late period of human evolution, where it appears now that we have 5, 6, perhaps even more different types, hesitate to call them species, different populations, different types of humans on the planet, and that at some point, anatomically modern humans, Homo sapiens, call them what you like, expand out of our African homeland, cradle of humanity, and move in, and they have some interaction with these people. And I wanted to try and capture the sense of excitement of the last 20 years in particular, when I've been working in this area, and the pace of change, as you so rightly say, has been extraordinary. And so I wanted to sort of get a taste of that sense of discovery and excitement and interest, not just from the perspective of scientists, but from the perspective of archaeologists working in the field, but also these great stories that you get in human evolution, these stories of luck and members of the public that get involved. You know, I mean, we've had this for many, many decades, of course. The discovery of some of the amazing painted caves in France, Lascaux, and, you know, sites like that, which were discovered completely by chance by members of the public. And some of the most important specimens that we have in late human evolution were also found by people who were just wandering around, not doing much, you know, taking a dog for a walk. A monk praying in a cave at Baixia in Western China discovered this amazing jawbone, which is the Baixia jaw, which is likely to be a Denisovan. So all of these things I wanted to try and capture and talk about in the book, and give that sense of excitement, but also luck that we also get in human evolution.
That sounds plenty of reasons to produce this great piece of work, and also luck is indeed something that we all know. It plays quite a role, but as you say, and you write in your book, having luck is a lot of work, right?
Yes, indeed.
So it's the combination of those two things. And so one of the things that comes through very strongly in your book is the impact and the effect of these new scientific approaches and what they reveal about our evolutionary past. So what is, in your view, the most important maybe of all these different approaches we can use today, and why?
Yeah, so well, we're in the middle of this omics revolution, genomics, proteomics, etc. And I think that genomic analysis has been the biggest thing. I mean, as I say in the early chapters, you know, I remember arriving here in Oxford in 2001 and meeting people who were working in the then very, very new ancient DNA laboratories, the Ancient Biomolecule Center. I worked with these people, and I was responsible for dating some of the sites that they were working on. And mainly they were working on animal DNA. And every time I broached the subject of human DNA, they'd say, "No, not going to happen. Impossible. No, never." And so why is that? Well, the contamination. You touch a bone, and you contaminate it, and it's human DNA. Impossible. And so, you kind of get the sense that it's never going to happen. But of course, you can always have this surprise element, not only in lucky finds, but also in scientific developments. And as we all know, over the last, particularly the last decade, starting in about 2010, the pace of development and technologies, chemical methods attached to ancient DNA work has proceeded at a real pace. And we've seen this massive exponential increase in the ability to extract demonstrably authentic ancient DNA from human remains, ancient human remains too. I mean, it's incredible when you think that we couldn't do this work before about 2005, and now we can get ancient DNA sequences. I wouldn't say almost routinely, but it's getting that way. In the right skillful hands, of course, and under the right laboratory conditions. So I think that ancient DNA has been the key thing, really, in my experience, of unlocking some of the big unanswered questions. But of course, as you say, it's not a technique that you use alone. You've got to use it with other methods and other techniques. And part of the excitement of working in these fields is that you work with lots of different experts and from lots of different disciplines. You can't just do it alone. You have to be a collaborative animal nowadays, don't you? And so working with geneticists and with experts in chronology and stable isotope analysis and proteomics, and increasingly, is a really important thing. But I think that if you look at the impact of this technique, DNA has been absolutely massive. We sequenced the Neanderthal genome, able to now determine whether or not we have interbred with different populations, increasingly with powerful statistical tools, being able to probe modern populations and modern human DNA to compare against those ancient sequences, to work out how and when and how often we interbred with archaic or disappeared cousin groups in the past. And also, intriguingly, as we might talk about later, to work out what in fact this DNA is and what it contributes to us today because of our health and the health of others.
Absolutely. I think a lot of people would agree completely with what you're saying. And it feels almost we are touching the sky in terms of what is possible today and what was possible even just 20 years ago. Even when I started my PhD, I mean, it was by then there was no sequence of, for example, the 500,000-year-old fossil from Atapuerca, Miguelon. Yes. I mean, that seemed completely, a miracle when happened. That such old fossil could be the subject of sequencing. So yeah, it's moving very fast, and I think you touched a very also important point that it's moving fast. Novel techniques are emerging with a lot more power and a lot more accuracy, but also the interdisciplinarity, I don't know if we can call it, but the teamwork is now vital in paleoanthropology. You cannot be alone, morphologist or paleoanthropologist looking at your assemblage of fossils. That is essential work that depends on a number of different expertise.
Yes, yes, absolutely.
So
and
so
it all starts, really, of course, in the archaeological site, doesn't it? It all starts with that very careful approach to archaeological excavation and the context of that material in the site. Without that, we're really lost. And so, the archaeological expertise is fundamental to all of this work. That is hard.
That's absolutely true. And in the accuracy and the detail and method used to dig today compared with also, what was done before. So I was thinking, as you know, I work in Africa in very old sediments. So nothing, I think, even though we want to be very optimistic, I think it may take still quite a long time until we can apply a lot of these techniques in some of the sites we work. But we don't have, for Africa, DNA preserving at all well prior to 10,000 or something years ago. So what do you think are maybe the scientific approaches we can expect to see playing a role in unlocking some of these aspects of our old evolutionary history? For example, I think we were talking today about, this DNA from sediments. This is a new breakthrough that, and I think I just saw a paper last week or 2 weeks ago where they even managed to get nuclear DNA now. So I'm thinking also, what can we expect, but also what should we be doing today to make sure that when new methods are in place, we can still make use of them and you minimize damage, right?
Yes, absolutely. So in Africa, it's a particularly acute problem, of course, because of the preservation state. And other parts of the world where, you have the combination of heat and temperature and water and the presence of bacteria and microbes. My friend Tom Gilbert says that DNA is just like ice cream, and in colder places it sticks, and you can eat it properly and over some hours sometimes, whereas in the hot places it just melts. And as the parent of a child, everybody knows that a child with an ice cream on a hot day is a rather difficult thing. And of course, when we look at Africa, and as you say, when we look at genomic data that's been extracted from specimens there of human remains, it's a very much more different picture than we have in the rest of, for example, in Eurasia, where we've got about 1,000 ancient human genomes of various age, and about 80 by comparison in Africa, of which a very, very small number are older than 10,000 years. And these come from places like Morocco in the far north, and sites that are slightly less prone to the disappearance of DNA. So I think one of the most exciting things recently that is, I'm sure, going to contribute to this greatly, and in fact it already is contributing, is looking at proteomics and proteins, because of course proteins survive much longer than DNA does. So if you're looking at the oldest ever DNA that's been extracted so far, it's something between 700 a million years, something like that. Whereas if you look at the oldest proteins that have been extracted, you're looking at something almost 4 million years in an East African ostrich eggshell sample, interestingly. So the thermal degradation histories of these two various biomolecules are much different from one another. So I think if you might recall last year, there was a fascinating paper that came out from the Copenhagen group in which they extracted proteins and proteomic information from the Homo antecessor specimen from the Atapuerca site in Spain. And there they obtained that proteome from a specimen that was dating to between about 780 to 950,000 years ago, or something of that order. And they managed to place it on an evolutionary tree basal to later hominins like Neanderthals, as we'd expect, Denisovans and Homo sapiens and so on. So these kinds of techniques, using much more long surviving biomolecules like the proteins, I think are going to have major implications for our ability to put things in the right place, as it were. And perhaps this is the future, and this is the way forward. It seems that these proteins can survive, although they're very low levels in archives like dental remains and teeth. And so perhaps this is the way forward if we're looking at material from Africa to try and understand more about the evolutionary history of hominins in Africa.
Sounds so exciting. Sounds like maybe even in our very old sites in Africa, maybe there will be something that we can soon
I hope
also
so.
start
That would be wonderful, wouldn't it?
That would be a dream. Yeah, and talking about Africa, of course, when reading your book, we come across some of these hot debates over the years regarding the data for the time of emergency of Homo sapiens in the issue from Africa to Eurasia. I mean, of course, there is consensus in terms of Africa being the cradle of humanity, but that even there, not for Homo sapiens, but for the origin of hominins, there is obviously controversy, but not for Homo sapiens. But in terms of these bitter debates, what is your view on this debate?
Yeah, so the out of Africa question, yes, it's a big area of controversy. My own view is that humans came out of Africa at various times, often, and it was probably influenced by several factors, push and pull factors in that continent. I think that there's now convincing evidence for much earlier movements of Homo sapiens out of Africa prior to what was for many years the sort of orthodox model, this 50 to 60,000 years ago out of Africa model. We have, for example, a very interesting find in Greece of the Apidima 1 specimen, which was published in 2019, despite the fact that it was excavated and found a long time before in the 1970s. And uranium series dates on that specimen suggest that it could be older than 210,000 years ago. That's getting near to the beginning of the lineage, really. If you think about modern Homo sapiens going back to, let's say, 300 to 350,000 years ago. Of course, it's very difficult to pinpoint this exact moment when we have people that look and physically like us, but certainly that's very early and quite convincing evidence. We've got Mislia Cave in Israel, where we have a human jawbone with teeth in it that looks very convincing. It was found in 2002, that dates to about 190,000 years ago. And then in China, we have some very interesting finds as well. Some are slightly controversial, but I think that another very convincing piece of evidence that suggests that there's a very early movement of Homo sapiens out of Africa is that in the Denisova cave Altai Neanderthal group, we have evidence there for the integration of Homo sapien DNA into Neanderthals, which is estimated to take place at about 100,000 years ago or earlier. So that to me seems pretty convincing. Similarly, when we look at some of the DNA that we have that are mined from modern living humans, we find that there's a vestige of very early DNA that seems to correlate to this very early period of 100,000, but the most significant is this later period, this 50 to 60,000 year period, where we have, I think, a larger group coming out of Africa, perhaps more widespread. And certainly these are the people that we see passing on genetic information to later populations in greater abundance, if you like. So I think that the answer to the question is that Homo sapiens came out several times. We're finding more and more evidence for this, but it was only later that the most significant out of Africa movement came about.
it's becoming so complex, the picture. So complex. It's you need to be on the top of this and follow. You need to really read permanently, be permanently updated, because there has been just so much information published.
Oh, totally.
I mean, you will know better, because you, of course, you did plenty of research for your book, but I mean, there are hundreds, maybe thousands of papers, and of course, everything relating to genetics, genomics is published at a velocity that is faster than normally other data sets. So it's really, really fascinating. But very, very fast forward in terms of rethinking the puzzle of the origins of Homo sapiens and the multiple dispersals within Africa, which is something
Yes,
that
we haven't mentioned that either. Yes, exactly. A great deal of complexity likely there.
Absolutely.
It's so hard to keep track of things. It's so complicated. I mean, this is one of the reasons why I really respect some scientists, like, for example, Chris Stringer. He seems to have this ability to be able to integrate all of this stuff and to keep track. And to make it look easy. I really struggle there.
No, but I think you don't, because your book also, does precisely that, makes it seem very, easy to follow all of these different avenues of information. And so continuing in a complex subject, and also very exciting. I mean, the theme of interbreeding, right, comes through in your book quite often. There seems to be a great deal of it going on. Now, this is particularly, I mean, it's quite interesting for me, because I mean, before I came to study in the UK and then stayed, I used to work in a small place in Portugal where the Lapedo child, if you remember that discovery,
yes, of course, yes.
was discovered. It was proposed at the time, right? Maybe you have the monography.
I've got the book somewhere behind me.
So remember that, it was proposed to be the product of interbreeding between Sapiens and Neanderthal. And so at the time, I followed really closely the very, very heated debate between Zilhão and everybody else, and Paul Mellis and everybody else. And at the time, we lacked a lot of the techniques and methods we have today. I suppose today one could give the final answer and close that debate, but what do you think in terms of the extent to which different hominin groups have difficulty producing offspring that could themselves go on to produce basically fertile offsprings? Yes,
it's a really good question. I think, yes, I remember that debate very, very clearly. And it seemed to me to be, I was erring more on the side that I didn't think that it was that Lagavelio, the child, was a product of an interbreeding event. It may well be that it was, because of course we have now evidence that this happens quite frequently in the paleoanthropological record. And I think what really amazed me, and I was quite taken aback actually, because of my initial degree of skepticism, was the Oase jaw, which was excavated in Romania. We dated that in our lab here in about 2006. And when the paper was published, Eric Trinkaus, who was the first author on that paper, he had noted that there were certain features on the jaw that to him, and in the teeth, that to him echoed Neanderthals. And so he put in that paper that he felt that there was perhaps a recent admixture that had resulted in this, and that there probably was some degree of interbreeding. And I was very skeptical. I was a co-author on the paper, and I kind of let it go because Eric is a big name in anthropology, and it was his work. So I said, okay, well, that's fine. I mean, it may or may not be true. And of course, subsequently in 2018, genomic data was extracted from the same specimen, which showed amazingly that 4 to 6 generations prior to the Oase man living in Romania 40,000 years ago, that he had a Neanderthal grandfather in his lineage. So we have this amazing evidence. So the question you asked was about breeding and whether or not these groups could then go on to have children of their own, and the possibility, therefore, that we might be looking at possibly hybrid populations. So when the first genomic data was being extracted from Neanderthals, and that was mitochondrial DNA, so this is the internal lineage of DNA, that showed absolutely no evidence for any Neanderthal DNA in Homo sapiens at all. And so to many, that was as much as we needed to suggest that there was no interbreeding that happened, and perhaps that even in places like Western Europe, Neanderthals and modern humans didn't even meet each other. It was only when the nuclear DNA genome, the whole genome was sequenced, that it became apparent that that wasn't true and that there was an interbreeding. And what's interesting is that in certain parts of the genome, that there are areas where you've got a bit of a desert in terms of Neanderthal DNA. One of these is on the X chromosome, and there are other parts that are also very free of Neanderthal DNA on parts that code for the testes, the male germline part of the genome. And so a lot of people have suggested that there may have been problems with Neanderthals and modern humans having viable offspring from one another. But more recent work has suggested that this might not actually be the case. A group here under the work researcher Gregor Larsen published data which uses a small part of the genome, this area known as cytochrome B. And this seems to be very informative in terms of predicting whether or not two different species can have a viable offspring. And they tested their approach using this cytochrome B or Cyt B method, using animals and also using different species of humans subsequently. And what they found was there was every expectation that humans and Neanderthals and Denisovans could have produced viable offspring and they wouldn't have any problems with fertility. So I guess my feeling would be that the separation in evolutionary terms between us and Neanderthals is of the order of 550,000 years ago, let's say. But we haven't been separated for that long, I don't think, in order to have these major problems with hybrid infertility. I think that we probably could have offspring. And the question then is, how regularly did it happen? And as we've seen, it seems as though it happened quite frequently, in places where they met.
Which is so interesting. I mean, certainly, sometimes I wonder, if we tend, as we do in so many things in human evolution, right, with our human way of approaching human evolution, we don't apply the same rules or the same principles that sometimes we apply to the rest of the living world, basically, because we do know in terms of hybridization that we have studies of lots of different organisms, from birds, from finches to trees, oak trees, showing that it can be quite a beneficial process, and it can help colonizing species to actually flourish in new environments, because it gives them useful genes, right? So it can be a very beneficial evolutionary strategy. So maybe that's why we are, as more and more discoveries are made, as more and more hominin species are sequenced, this is becoming more frequent than what we thought it was.
Absolutely. I mean, there's some really cool things that are happening. I mean, there's a fantastic paper that came out recently by the Leipzig group by a researcher called Ben Peter, and he looked back at all of the genomes from Denisova cave, Neanderthal and Denisovan, and he used this new powerful statistical technique to work out the degree of intermixing between these various groups. And he showed that every single hominin remain that has so far been sequenced has evidence for not just one interbreeding event in their past, but sometimes 2 or 3 in their deeper past, and small sections of DNA that have the vestigial remains of this. So hybridization is something that is happening, I think, a lot whenever these groups meet, but it can't be the case that they're single groups, because I think the DNA is separate enough so that we can tell if they're Neanderthals or Denisovans. There's obviously isolation going on, but periodically they come together and they meet, and when they do meet, they hybridize. And as you say, I think this hybridization is so exciting. I think it's the key to understanding a lot of things in the way that different groups adapt to new environments. And there's been some really fascinating stuff which marries the physical appearance of hybrids, genetics. And, I mean, I talk about this in one of the last chapters about baboons as a for looking at the genus Homo. So in Africa, we have about 6 different baboon subspecies, don't we, that they live in parts of Africa. And then we have these amazing sort of hybrid zones between the population, which you're the expert. I should stop talking at this point.
No.
And so the genetic analysis of these various populations has shown that these groups interbreed with one another, and they produce hybrid offspring that have often strange physical appearance. They have, you know, slightly bigger teeth in many cases, huge teeth. And Rebecca Ackerman from Cape Town has written a lot about this, and her work is
that's right.
phenomenally interesting, I think. It's really, really amazing because it shows that there are sort of curious things that happen amongst hybrid groups, and these hybrid zones. And, you know, in Denisova Cave, which, as you say, I worked on quite a lot, you know, it's halfway between a Neanderthal-dominated west of Eurasia and a Denisovan-dominated perhaps east of Eurasia. And so it may be that it's on one of these kind of hybrid zones, and where they overlap sometimes, they interbreed with one another. And what's cool about it, I think, is that if we look at hybrids as producing sometimes these strange geomorphological, these strange morphological features, like big teeth, for example, well, some of the Denisovan remains we've got at Denisova, Denisova 4, the tooth, it's huge. It's a massive tooth. It's of the size of something much older in evolutionary time, you know, 1.5 or 2 million year old time. So could this be an artifact of hybridization? And also, the other thing that we're finding out a lot is that hybridization can confer adaptive advantages and benefits genetically. So we're finding that a lot of the interbreeding that took place between Homo sapiens and Denisovans resulted in the integration of genes which confer benefits in terms of immunological response and the ability of groups in tropical environments to resist disease from those environments. So it's almost as though we're piggybacking on the slow evolutionary adaptation of different groups, different environments. We're borrowing the DNA from them, and there's an advantage. So that DNA surfs almost through the population.
Absolutely. That we are alone now, but we got a lot of help to get here, basically. I don't want to, you know, hijack by now talking about baboons. You're completely right. And just to say, I mean, that's one of the reasons why we're doing so much work now with the baboons in Gorongosa, is that we think, and the preliminary data is saying that what we have in Gorongosa is showing a mixture between two species, the Shackman, the yellow, and we are right 100 kilometers south from one of the hybridization zones. Amazing. And so, I think that can help, you know, shed some light. And of course, this data is also consistent with the data we have for other species. I mean, we have similar data for chimpanzees, for bonobos, for the hypothetical extinct pan species, ghost that you also talk about. But I want to shift gear because I can see the time passing, and I know everybody wants to hear about the Denisovans. And you mentioned briefly, you know, they are such a cool mystery, and they are they are not as known as Neanderthals, not at all. We still know so little. So what can you tell us about, you know, who were the Denisovans and what is more or less their distribution? What do we know at the moment?
Yes, so as you say, it's very early days in terms of Denisovans, but we have some really intriguing hints about them and who they were and what it looks like. There was a lovely paper published a couple of weeks ago which confirms this idea that instead of one Denisovan population, it looks as though they have a much more structured population and a much more complex history. For example, we think Denisovans and Neanderthals are separate. Well, there are two populations of Denisovans at least that have a basal separation that is of the order of the same time as Neanderthals and Denisovans. So we have groups that are as separated from each other from Neanderthals within the Denisovan lineage. So that's amazing. And I think that what's likely to be the case based on some of the modern living human DNA that is being analyzed, again, with these powerful statistical techniques, is that we've got several groups of Denisovans that have become isolated from one another. And it's likely, I think, that there's one group that lives, or that lived, to the east of Wallace's line. So that's in islands Southeast Asia and places like the Philippines, Papua New Guinea, island Melanesia, Aboriginal Australia. I think that's likely that there was a Denisovan population that lived there. And also in eastern Eurasia, in places like China, we have at least two populations there, and probably more. And we also have evidence that, that these various groups at various times interbred with anatomically modern humans in these various places. The recent paper that was published a couple of weeks ago suggested at least 4 different integration events that took place. And intriguingly, one of the most recent could be as recent as 21,000 years ago, which suggests that Denisovans survive for much longer than Neanderthals did. We think Neanderthals probably disappeared around 40,000 years ago, at least where we have good data. But Denisovans appear to have been more successful in the light of the undoubted appearance of Homo sapiens in these areas. And that may be indicative of the fact that in island Southeast Asia, it was possible to become more isolated. There were groups living on islands, perhaps, rather like the hobbits and Homo luzonensis, that were able to flourish for quite considerable periods of geological time. And so, yeah, the Denisovans seem to be more diverse genetically. They seem to be multiple populations, but we only have these tiny vestigial remains. The physical remains are so few. We really need more archaeological excavation in these places to find Denisovans on the ground. More and more.
But already very, very, important information, very interesting, and quite a wide distribution for Denisovans already. Yeah. In talking about Denisovans, I mean, one of the most relevant and exciting parts of your book, in light of the interbreeding concerns, in light of the interbreeding concept, concerns the discovery of the so-called Deni, the history around finding Deni. Could you tell us more about this finding and why is so important?
Yeah, I mean, this was one of the main motivations for writing the book, was to share this exciting story, and it was a fantastic thing to be involved in. And the idea for looking at this came from the fact that at Denisova Cave, we have an archaeological site there that spans 250,000 years, but the bone that's found in the site, as well as being very revealing of genetics, the bones are tiny and they're fragmented, and 95% of the bones from the site we can't identify because they're tiny pieces of bone. I've got some bags of the bone from over there, but anyway. So they're tiny little bone fragments, and we can't identify the species. And so Katerina Duca, my research partner and wife, and I were at the cave in 2014, and we had this sudden realization that what we could do was to apply some of these new peptide screening analyses to try and screen thousands of bones to find human remains. And so this technique called Zooms was developed in York by Matthew Collins and Michael Buckley, and it was sort of developed in the early 2010s. And it hadn't been used in this manner to look formally for human remains before. Anyway, so we got permission to do it, and then a fantastic graduate student called Samantha Brown took on the project, and she screened hundreds and hundreds of bones. It wasn't until number 1,227 that we found a hominin bone, which was stunning and very exciting. And this is a 3D scan of the bone and what it looks like, so you can get an idea of the size of it. It's about 2.5 centimeters high, tiny little thing. And this bone was then genetically sequenced. Its mitochondrial lineage was Neanderthal, but the nuclear DNA stunningly and very surprisingly told us that this was the remain of a girl who was a 50-50 hybrid. Her mother was a Neanderthal and her father was a Denisovan. And so, I mean, I still get goosebumps even thinking about that, because it just demonstrates again the power of genomics, the power of this technique to reach back in time to 120,000 years ago and to put together these family stories of these ancient humans, and to work out that people were living at the cave at the same time, they must probably, and that they interbred with one another when they met. So it was a stunning realization and a stunning development, I think, in the story of this these early humans.
It is so, yeah, it's so exciting. It is very, very exciting. All this work is very exciting. And something else that you also talked about before, and I'm going back to it, because you talk about the relevance of this genetic exchange for the living people today, right? So can you say a little bit more about this? I mean, we generally know about our genetic legacy, what we inherit from these ancient relatives of ours.
Yes, I mean, I think this is another area that is very exciting and developing. What is the function of the genes? What is the function of the genetic exchange that we experienced? What does it give us? How much of it is there? And latest data is showing that there are some quite big differences between what we inherited from Neanderthals and what we inherited from Denisovans. Denisovans aren't so well known because we don't have very much in the way of high coverage modern human living genomes in order to compare with. So in places like the UK, we have these fantastic biobanks, and the biobanks contain high coverage genomic sequences and phenotypic information that are gained and gathered by surveys. So they interview people and they say, so, you know, what are you right-handed or left-handed? Oh, I'm left-handed. Do you like getting up early in the morning or later in the morning? Oh, I like getting late. So they have all this survey information, and then it's possible for scientists to go through and look at the genome and the genetics to figure out what might be significant in terms of explaining those phenotypic data sets. And about 10 years ago or so, Neanderthal DNA and allele sequences were added to the data set so that scientists could then start to look at what is significant in terms of the Neanderthal parts of those questions. And all of this data has been subsequently analyzed. And so we now know that Neanderthals often gave us things that are beneficial, but they also gave us things that are slightly negative. Like we have DNA that codes to type 2 diabetes that we inherit from Neanderthals, smoking behavior and smoking addictions from Neanderthals, things like lupus. We also have some positive things about the type of quality of the keratin and the hair, skin that we obtained from Neanderthals too, and some other funny things to do with, you know, chronotypes. That's the type of day you like. So if you're a morning person or an evening person, it's quite highly correlated with a Neanderthal allele. For Denisovans, a lot of the data that we get now suggests that immune response is very closely linked with Denisovan DNA integration. And because a lot of the Denisovan DNA that we find in living people is in places like island Melanesia, Papua New Guinea, Aboriginal Australia, and so on, and to a lesser extent in Eastern Eurasia, and a lot of these immune alleles and immune gene variants that we seem to get, it's likely that I think that these were very highly present in these populations because it conferred this advantage that we talked about before. It meant that people were able to resist the tropical array of diseases, and immune response is of course a very important thing when we're talking about the ability of people to fight illnesses and diseases in these places. So we're getting more and more information all the time about what we inherited. Some of it's related to COVID even. There was a paper recently published about the effect of Neanderthal DNA on certain parts of the genome with respect to COVID. And it turns out there are good things and bad things again. So it's complicated. And the functional analysis of this material is an active subject of investigation.
That's one of the most fascinating things. And that's, I think, also where the public identifies
yes, exactly.
You know, concrete links between what one is doing when one is digging and doing all this work to try to reconstruct the past, and actually some real benefits.
Yeah, it's amazing,
potentially to avoid our extinction in the present.
Yes, exactly.
You know, and battle pandemics and all of that. So it's really, it's incredible how far, you know, how wide and how far are the links and the potential of archaeology, paleoanthropology as a field of research. I'm going to move to some questions that are, you know, more, maybe I think the audience would like to know something about it, because I can see the time.
Yes.
It's going so well. I'm enjoying this so much. I really don't want to stop, but maybe I can ask you, what are the big unknown remaining questions in human evolution for this time period? So we are talking about 300,000 years ago. And are you working on some of these now? I mean, what are the...
Well, I mean, I think one of the big questions I have in my mind is, you know, what is the distribution of Denisovans, and what is their technology? What is their adaptation? Because we only really have 2 sites in the world where we have Denisovan remains present. That's Denisova Cave, the eponymous site, and at Baishia Cave in the Tibetan Plateau. And so we have this intriguing data from modern human living DNA, which tells us of the distribution of Denisovan DNA now. And I think that, you know, what really I want to know is, you know, where were Denisovans living in this period from, you know, 400,000 years ago to let's say 20,000 years ago? What were they doing? Where were they? We've got lots of physical remains of let's say archaic Homo sapiens that we don't know yet are Denisovans. Are they Denisovans? That's another major question. We want to know this. genetics and proteomics will undoubtedly play a big role in this, particularly in places like China. We've got a lot of human remains that we don't really know what they are yet. I think for me, one of the keys to unlock this is this new emerging field of sediment DNA. You mentioned this before, the ability to extract DNA from sediments. I mean, it's almost like science fiction. I couldn't believe it was ever going to happen again, rather like the modern story of DNA analysis. But using these techniques to actually core down into sites and to visit old sites and to analyze the DNA there and see whether or not we can identify the distribution of Denisovans over time is a really interesting possibility. And I'm really excited by that. I'd love to get involved and do some of that work and go and test that. We've done some of it, and we talk about this. We did find some human DNA, but the techniques now are getting so much better, and they're using robots to sort of produce more and more of these sequences in the lab. So it's hugely exciting. So I think that's one of the most interesting questions for me, is the distribution, and also what they were doing in these places, because Denisovans living in the tropics, if that's what they're doing, now that's a difficult place to live. That requires technology of a sort that we often associate with early anatomically modern humans, moving into places like Sri Lanka and India and the subcontinent, and into places like Indonesia and so on. But Denisovans must have been there, if our hunch is correct, they must have been there for quite some time. So they must have had a degree of technology to live in places like rainforests. And so that's another really exciting thing to look at too.
Absolutely. And I mean, and also the possibility to disentangle some of the questions we have around, taxonomy versus, taxonomic variation versus behavioral variation, right? It's a huge issue because, of course, maybe not so huge, even though it is in the case of the Denisovans, but not so huge as you come to recent times in human evolution. But for older times, yes. I can think of another period that about 1.4 million years where you have 4 hominins at the same time. You have Homo erectus, Paranthropus, Homo habilis, Homo rudolfensis, all in a relatively small area. And we are not talking enough about, material culture. And we find, if you find tools in a site where you have Homo and Paranthropus, that's described as being Homo. But actually they could also be Paranthropus, and how do we know, if you don't have the hominins associated, how do we know which assemblages and which morphotypes belong to, because we have a lot of assumptions at the moment in terms of this is the morphotype or the technocomplex used by Homo erectus, say. And so I think this, it may be possible to do something, because there is a lot more of everything. There are a lot more sites, there are a lot more assemblages, a lot more, relatively speaking. It may be in the most recent times that we are going to be able to disentangle a bit more of this,
Yes, absolutely. I think that's so true. And linking different human groups to different types of stone tool industries and material culture is a massive thing, because as you say, we do rather assume a lot. And now that we have the possibility that there might be hybrid humans running around, it becomes even more untenable and difficult to say, oh look, I found this specific type of stone tool, that must mean it's Neanderthals, or that must mean it's
absolutely. And then we drop in, and then we throw in all the tools from non-humans with that, and then it makes a huge no. But so still, can I ask you to do some prediction of the future? What do you think are the most likely locations for the next hot discovery in this field?
Oh God, yes. That's a tricky question. I mean, one always thinks. I mean, Becky Ragg Sykes. She I think it was her who mentioned this, that her dream would be to have, in the permafrost areas, you find, complete frozen bears and mammoths and so on. Wouldn't it be amazing to have a complete, or a frozen Neanderthal, or perhaps a Denisovan? I mean, that would be incredible. I mean, it's possible, I suppose. I think, there's a lot of evidence for these so-called ghost populations in some of these ancient genomes, and it looks as though there may well be more human populations and lineages that we know now. And certainly, I think in island Southeast Asia, there's a possibility that we could well find other groups that may have interbred with us. There's a big question about Homo erectus, which I talk about. How long did Homo erectus last in island Southeast Asia, and was it last was it late enough that anatomically modern humans could have perhaps met them? So I think sort of island Southeast Asia seems to me, and Papua New Guinea and places like that seem to be very exciting for me anyway, because I think that there are great chances there that we could discover some really profound things about how humans adapted to these environments and when, and perhaps even find new lineages that we didn't know existed before. So I think that's.
I think you're probably very, very right. You just need to find researchers that want to work in some of these. I know a few.
Yeah.
I suppose, because this is such a fast-moving field, like we said in the beginning, you know, where publications are out every day, that when you were writing these, were you worried to some extent that you were writing your book and then it was going to be outdated? Because I worry about my lectures all the time. So writing a book, and it's so great that I think the book manages to, you know, to overcome that. Many ways.
Yeah, thanks. I think, yes, of course you are. You were always worried that next week a Denisovan skeleton might be found that dates to 5000 BP. Oh, I don't know, I'm just joking, but there's always that. But fortunately, I've managed to, well, I don't think I, you know, asked around and asked for any information ahead of publications of specific papers. I didn't do that in many cases, but I think I was careful and cautious in some areas to say, you know, this could be and that could be, and, you know, to cover myself just in case. But luckily, so far, so good. I think I've finished writing the book in November last year, and luckily, so far, there has been some stunning discoveries, but nothing that's really overturned what I've said.
Absolutely, and I think in the way you present things, definitely it buffers. I don't think it's going to be outdated easily. You know, of course, there will be a lot of new things. Another book can come after this one, but
exactly. But of course, you know, I'd be so happy if something did come along to say something new. I mean,
of course,
to just quickly.
That's what we want. That's what we want.
Exactly. So
okay, there has to be a pandemic question.
Right.
It's mandatory. So I hear that you wrote the book, you know, mostly during the pandemic. Or a good portion of it was during the pandemic. Was this, you know, did it make it more difficult to write a book? Or
so I was quite lucky because we spent most of 2020. I was at home in New Zealand, and then I spent most of the rest of the time in Greece actually, because we were very luckily able to go there and stay with Katerina's family, and so. I spent a lot of time in the early morning hours from about 5 a.m. till about 8 a.m. working before the rest of the family and the children got up. So this was a book that was produced in the very early hours. And of course, that has its own advantages. Fortunately, I'm a morning person. I think that might be Neanderthal DNA related. But whatever the case, it was great. I really enjoyed writing it because it was a great opportunity to look at other papers and read around the subject a lot. And I think any academic should at least once have a go at this and write a book. I thought it would be a little bit more simple than it turned out to be. But I moved into areas that weren't very familiar to me, and so I did have to do a lot of work, and fortunately talked to a lot of colleagues who were able to help me. So in the end, I think it was a great experience. I'm just glad it's over.
You certainly did an absolutely outstanding job. But you touched about important point, because I think a lot of us have not yet written a book, and they are kind of scared of doing it for many reasons, or postpone it, or think I don't have enough to say. What advice do you have for other academics thinking of writing a popular science style book? Because that would be, you know, if I would go to write one soon, it would be also, I would like to be able to write to reach a popular audience too.
Yes, I think it's great. I mean, we're scientists, we need to reach out and share data and share results. And that, as I said at the very beginning, that was part of my motivation, was to be able to say, when people asked a question, well, you know, you could read this book or that book, and this might help you and might be interesting to you. So I think it's a really good thing to do. So what I did was I asked some of my colleagues who'd written popular science or popular books to see what their advice was. And they said, oh, you should try and get an agent, because an agent will help you to advise you as to how to go about getting someone to publish it, because, you know, I had no idea about this. I'd never done it before. So that was really good advice. So I enlisted the support of a publisher, of an agent, I should say. And she was able to give me advice on what to do to put together a proposal for writing a book, and then to help me to pitch it to various publishers. And it was getting to that stage that was absolutely invaluable. And so I would first recommend that you do that. And then if you were fortunate enough to get to a good publisher and you get a good editor, well, that makes all the difference. I mean, I had this fantastic help from this guy, Connor Brown at Penguin at Random House, and he was super. You know, he gave me really good advice, and the team there were just wonderful. So if you're lucky and you get to work with some good editors, I mean, it makes a huge difference to writing a book, I think.
Thank you so much. Thank you so much for, this was absolutely wonderful to have the opportunity to discuss with you and to hear in more detail the many different exciting discoveries and work going on that you reveal in depth in your book. I think I would call, if possible, Robin to give us some help, assistance here, because I cannot see questions here. So I don't know if we have a few questions that we can still, oh, here is one. Okay. Would Homo sapiens mothers experience a more difficult birth due to narrower pelvis, or were there more births from Neanderthal mothers? It is quite possible. Any percentages? My burning question.
Yeah, that's a very good question, and there have been some analyses of pelvic shapes and so on, and particularly done the work of analysis of Neanderthal neonates to look at their size and the birth canal and so on. And my understanding from this work is that there isn't a great deal of difference between the two, perhaps a small amount, but that might have actually been a significant enough difference between the two to perhaps make a difference, even a small difference in the ability of different groups to have live births might have made something of a difference in terms of their evolutionary success. I'm not really an expert on this though, so I don't feel as though I can really comment, but I think that if the person asking the question were able to do some searches for some of the publications that have been undertaken on these analyses of Neanderthal neonates and the birth canal and the pelvis, that might be a good thing to do. But I feel as though I'm not really an expert in that area, and I can't really speak with great authority on that.
Thank you very much. Thanks. I think there is a lot out there, and I think also that we probably don't have the answer yes or no to these. I recall there is a paper out maybe even last week from Nick. The pelvic floor.
Yes.
Sorry, the pelvic floor. So it may not be all just around the morphology of
yes.
of the
yes, that's a good
pelvis.
point. Yep.
And I think and I don't think we have enough information to say much more here. It's not also my expertise in terms of Homo sapiens.
Yes.
But do we have any more questions from the audience? Otherwise, I will ask if we have. Robin is going to let me know. Otherwise, I will, if you allow me, just ask the $1 million question, which is if you could dream up on one major discovery in this field to be reported in the next 2 or 3 weeks, what would that be?
Goodness me. Well, I think I'd go back to, I think I'd probably go back. God, that's a really tough question. I think I probably would really like a skeletal remains of a Denisovan, and very well preserved remains to be excavated in an unimpeachable context, within which we could also see evidence for their material culture. Perhaps in an archaeological horizon, an archaeological layer which has evidence from what they did and what they were doing. I'd love to see the physical appearance of a Denisovan. I'd love to see what kind of tools they were making, perhaps in more detail, and whether there were any organic artifacts that they were using as well, bone tools and perhaps even material of another nature. So I think that would be a dream come true. If anyone out there has found one, could you email me privately? I won't tell anyone, I promise.
I think this is wonderful, Tom. I can see there is another person, colleague asking, is it possible that caves will be revealed by retreating glaciers that could lead to Denisovan discoveries?
Yeah, that's very possible. And in the same way that we get the reveal of animals that are found from Pleistocene times, you know, like frozen remains of mammoths and baby mammoths and wolves and bears and things like that. There have been some incredible discoveries over the last few years, as sadly the permafrost begins to melt. And the corollary of this is, of course, that climate change is really happening now. I remember when the Iceman was found in the Ötztal Alps in Italy, and the discovery there was quite stunning. But we're going to find, I think, more of these things. In Norway, for example, we've seen lots of Viking artifacts have been revealed by melting ice and melting snow. And it's undoubtedly the case that in parts of Siberia, permafrost melt is also happening. So we may well find something up there, and that would be just amazing, wouldn't it? I mean, to find something like that. Incredible.
Oh, that would be absolutely fantastic.
Yeah.
So I'm just going to ask one more question. We are way, over time.
Yeah.
I think we have been here now for maybe an hour 10.
Oh God.
At all. But we have a few questions. I'm going to pick one and ask the audience to understand that I think Tom is doing so much outreach events with the book, so that what is not answered here today, it's going to be answered afterwards for sure. But somebody is asking, we heard a lot about molecular advances. Could you comment about advancements in more refined radiocarbon chronologies?
Yes, of course. So there have been several of these. Our ability to date decontaminated bone, I think, is one of the most exciting and one I've been involved with quite a lot. So we can now actually, instead of taking the bone collagen that we traditionally extract for dating, which is about 20% of the entire bone, and then dating that, what we sometimes find is that there are contaminants that are unremoved by the chemical methods that we use to get the collagen out. And so now it's possible to extract single amino acids from the collagen for dating. And we can extract an amino acid called hydroxyproline, which is very specific to mammalian collagen. And it means that all of the contamination is basically eliminated, except in times when somebody in a museum has used some animal bone glue to glue back some bones together, which is bloody annoying. Sometimes means that we get a problem there. And in fact, a few weeks ago, we published this paper on this new Czech skull, which did indeed have some cow glue that was used in it, which messed up our dating. So, but most of the time it's fine. So that's really exciting. And the other thing we have just since the beginning of this year is the international calibration curve that now extends the ability to correct radiocarbon dates into calendar years back to 55,000 years ago. Wow. So these two, I think, are two of the most exciting methods that mean that we can now date things much more reliably, accurately, and precisely than we could before.
This is wonderful. I think it's a wonderful way to end our discussion today. Thanks. I'm going to again really recommend this book to everyone. And I think all students must read this book, certainly all students that are having classes with me. Tom, thank you so much. Super congratulations for Thanks, this Susanna. Amazing achievement.
Thank you.
And it was an honor for me, a pleasure today to be able to discuss the book with you. I hope our audience enjoyed. As I said, please don't be sad if we missed some of your questions. Tom is having plenty of interviews, events. I'm sure he will be also happy to answer if you send him any question to answer.
Email and Twitter.
Yep. Absolutely. And thank you for attending today. This was a wonderful session. Thank you so much.
Thank you, Susanna, and thank you everybody for coming. I really appreciate it.